So what I have is a copy of the specification close at hand. Just going through the specification, and I'm going through point by point and just putting all the content in some kind of structure. Okay, and by the end we should have ourselves something that looks a little bit like a glycogen molecule, lots of branches. The main areas of biological molecules, which I'll put the main areas first and then we'll go into each one. The first major molecule that we look at is water, and then we're going to have essentially our macromolecules, i.e. our big molecules. Now, in terms of macromolecules, there's a concept that we're going to keep coming back to, and we will look at that when we get into the individual topics. But this idea that macromolecules are built generally with condensation reactions and are broken down back to their monomers as a result of hydrolysis reactions. Okay, so this concept of we have the monomers and they are kind of interconverted with their polymeric forms or converted into polymers. The process that converts the monomers into polymers in biology, it is always condensation. Yep. And the process that takes us from polymers back to monomers, and in many cases that is required in the body, is hydrolysis. All right, so this is a unifying concept. It's common for all of our molecules, and so I'm putting it higher up in the structure. Okay, but from this idea, then we're going to go into our individual molecules. Proteins, no, lipids, and in fact, might just take care of lipids down there, which leaves us this area for proteins. Yep. And then finally, we've got a little bit of testing for our biology. Oh, wait a minute. Nope. We also have mineral ions, etc. So we'll take care of that. Those aren't macromolecules, so we've got mineral ions. Mineral ions we'll have here, and then we've got testing, right? Practical. Let's just call that practical. And in practical terms, the two main things we need to be able to do with our biological molecules is A, testing for presence and concentration. So presence would be like the qualitative test, testing if something is simply there or it's not there. Concentration being quantitative, being able to say not only that something is there or not, but how much of it there is or how concentrated it is. And there's also a bit on separation. Okay, so we've got separation of mixtures of biological molecules, and in that way identification, but yeah, okay. So these are our main areas, and so I will highlight them as such, and hopefully I won't need to do too much reorganizing. Yeah, it's going to get pretty dense, but it is biology. We're not surprised by that at this point, hopefully. All right, so let's get into it then. We begin with water. The pattern that usually we follow is as so. So water, we need to know about its structure. First of all, structure. So in terms of structure, we have the oxygen, 2 covalent bonds, 2 hydrogen atoms. So covalent bonds, shared electrons. Remember that the oxygen is partially negative and the hydrogens are partially positive, so it's a polar molecule. But so the structure is that oxygen is covalently bonded to two hydrogen molecules. It's a bent molecule, or it's a bent arrangement of the bonds, and that is essentially what results in its properties. Okay, so what I would like you to think of is this: that the structure of a molecule, know the structure leads to the molecule's properties. Okay, so the properties of water are that it's a polar molecule, or that might overlap with structure, but these are somewhat man-made distinctions. You know, one thing kind of bleeds into the next, doesn't it? Properties are that water is polar, it's a good solvent. Well, actually that's a function, isn't it? Right, so it's polar, so we'll take solvent off there, that's more of a function. Properties: polar. Properties are high specific heat capacity. Interesting properties include the fact that when it freezes it's less dense than the liquid version of water. But again, we're not going to go into the detail here, but I'm just pointing these things out so you don't miss it or overlook it in your revision. Okay, so freezing properties, thermal properties, polar, cohesive, adhesive. Okay, so those are the properties. So water has its structure, the structure causes the properties, and the properties allow the molecule to have relevance, biological significance for life. And essentially that's what we'll call the function. Okay, so cohesiveness, so I'm just going to do some of these just for context. But it's not going to be an exhaustive list. So in terms of cohesiveness, can we think of any biological phenomena from any part of the specification that might be connected to water's cohesiveness, right? So we are talking about things like transpiration. Okay, the transpiration stream, cohesion tension, and that's connected to cohesiveness. The fact that it's polar allows it to be a good solvent. Properties, adhesive, freezing means that it could be a good habitat for terrestrial organisms, but it also allows aquatic. So in terms of habitat, it's not only good for terrestrial organisms, but it's equally it's good for the aquatic organisms because it's insulating for the body of water underneath the ice sheet. Okay, so and in terms of adhesive, well, the transpiration kind of doesn't happen unless the water can stick to the walls of the xylem vessels. So there is that. The idea of cohesive is also related to mass flow, right? Specific heat capacity is connected to homeostasis, and the fact that it helps organisms aquatic organisms to be in a more thermally stable environment. Right, so thermoregulation. But you get my point, right? And what we've done for water is essentially what the whole topic is all about, right? We got to understand the structure of these molecules. We got to understand how the structure impacts on their properties, and it's ultimately their properties that allow them to perform the functions that they do at a higher biological level. So moving from the chemical to the biological level. Okay, so that's water, I believe. Transport medium, coolant. Yeah, and so we move on to next the macromolecules, and we'll begin with the carbohydrates. How am I going to do this? Okay. All right. So in terms of the carbohydrates, in terms of the right. Okay. Now, what I've done is I've said, so this becomes quite straightforward at some point, but we're just going to put it here anyway, because I do see questions about this quite a lot, and it's something I take for granted. But now, the elements that each of these things are made of, so which elements are present? Okay, so in terms of carbohydrates, remember it's they're hydrates of carbon. So okay, now what I've done is I've said, so this becomes quite straightforward at some point, but we're just going to put it here anyway, because I do see questions about this quite a lot, and it's something I take for granted. But now, the elements that each of these things are made of, so which elements are present? Okay, so in terms of carbohydrates, remember it's they're hydrates of carbon. So the formula CnH2ON, right? So generally the carbohydrates will have this chemical formula, and which also indicates that they are made of carbon, hydrogen, and oxygen elements, and the atoms in those ratios. Yeah. Okay. Ring structure, glucose. So once we've done that, then we'll move on to what the monomers actually are. All right. So what are our monomers? So that we've got some specific examples of carbohydrates that we go in depth into in the topic. They are alpha glucose and beta glucose, and that's it, right? Alpha glucose and beta glucose, right? We need to know their structure. What I've done now is to kind of rearrange it, and I'm much happier with the new arrangement. And so we've got the structure of glucose up here, alpha version, beta version, and now what we're going to do is we're just going to discuss the different polymers that we can make from alpha glucose and beta glucose. Polymers are made from the condensation of the monomers, so we're very quickly just going to remind ourselves about the condensation reaction and the fact that we produce a glycosidic bond from the condensation of two sugar molecules. Okay, so we have an OH here, and we might have another OH group from another monomer. All right, so we've got two OH groups on adjacent monosaccharide molecules. Yeah, that's the first time I've used that word, isn't it? Right, remember these are monosaccharides. Okay, so we've got the two OH groups next to each other. Should be straightforward. hydrogen from one glucose combines, or the OH group from one monosaccharide combines with the hydrogen and oxygen from the OH group of the other monosaccharide. In doing so, a water molecule is produced, and when that happens, we lose the two hydrogens and the oxygen as they've become water. And what happens is that the oxygen from one of the monosaccharides now links via two covalent bonds to both monosaccharides, links the two together, and that is what is known as the glycosidic bond. Okay, so don't forget that. Monomers undergo the process of condensation producing glycosidic bonds, and just remember that COC arrangement, and you should be okay with that. Now let's move on, right? So the types of molecules that we can make are a few, okay? So from the alpha glucose side, from alpha glucose, we could have two. We could have starch, yep, and we could have, so these are alpha ones, we could have starch, and we could have the other alpha, which could be glycogen, whereas with the beta, we could have cellulose. All right? Okay, now in terms of starch, remember that the starch starch has two types of polymer. It has amylose and it has amylopectin, and the amylopectin is the amylose, let's just begin with amylose. The amylose is an unbranched molecule, right? So the glycosidic bonds happen essentially in a line, right? But that molecule is coiled, all right? So avoid saying straight. An unbranched amylose is unbranched, but it is coiled, okay? In terms of its structure. Right, so we do have unbranched and coiled, and the glycosidic bonds are from carbon number 1 to carbon number 4. So these are 1,4 glycosidic bonds. 1,4 glycosidic bonds. That's kind of all structural information. All right, so that's the structure. But what properties does it lead to? Well, it is quite compact. Yeah. And I think that's really all we can say about amylose, is that it is a compact form of starch. Oh, actually, because it is a polysaccharide, and it's going to be true for all the polysaccharides, while we didn't discuss the properties of glucose. While the monomers are soluble, the polysaccharides are insoluble, and that's an important reason for their existence. So insoluble. Okay, and because it's compact, you can store lots of glucose, and because it's insoluble, it does not affect, the water potential, and that's an important feature of storage polysaccharides. Okay, hope I'm not forgetting important things here. So one function is to store lots of glucose for respiration when it is required. Maybe we don't have a great external supply of glucose to a cell. In that case, the cell can start breaking down its store of the polysaccharide to supply the glucose for respiration. Okay, but in order to store lots of glucose, it wouldn't be ideal to just have lots of glucose, because that would lower the water potential. Water would come into the cell by osmosis, cause the cell to burst. Therefore, it's an important property of these polysaccharides is that they are insoluble and don't affect the water potential, meaning we can store a large amount of, we can store more glucose then. Okay, so there is that. Amylopectin, then, amylopectin is branched, but it's not highly branched as glycogen is, so that's like a contrast with that. Starch. Yeah, amylopectin is branched, and because of that, structurally speaking, it has 1 to 4 glycosidic bonds and 1 to 6 glycosidic bonds. And I hope at this point you know what that means. If you don't, have a quick look at the structure, find where carbon 6 is on the glucose molecule. It's right there. Okay, so it's connecting this carbon of one molecule to this carbon of another molecule. Okay, and in that way we can produce a branched polysaccharide, which functionally, or kind of properties wise, it makes it a structurally it makes it a branched molecule. But how does that affect the function? Well, I think that means you can store even more glucose storage, and it results in a higher rate of hydrolysis, because enzymes have more branch endings at which to carry out the hydrolysis reactions. Instead of having to chop one by one, they can multiple carbohydrate chains can be hydrolyzed at once. Okay, so there is that. I hope I'm not forgetting anything important there. All right, just to note that, starch is a plant polysaccharide. So this is happening in plants, whereas glycogen is what we are talking about in animals. So the animal form of alpha glucose storage is glycogen. All right, so animal right there. There's not a lot of difference structurally with amylopectin, so a lot of the structural information is similar. It is branched, or actually we'll call it very highly branched. Okay, it's very highly branched, so it's got 1,4, 1,6, just the same as amylopectin, but because it's highly branched, it has an extra high rate of hydrolysis, which evolutionarily makes sense because animals are much more metabolically demanding than plants, hopefully. Okay, take a look at your life. Highly branched, so you got a higher possible rate of glucose release. Now be careful, don't refer to polysaccharides as providing energy. Refer to them as basically providing the glucose for the energy to be released later by the process of respiration. Okay, so the process of hydrolysis doesn't produce energy. The process of hydrolysis produces glucose, which can then be used for energy if it goes through the process of respiration. So higher rate of hydrolysis. Okay, so if that's amylose, that might be amylopectin, but that would be glycogen. Okay, so while amylopectin has branches, think of glycogen as its branches have branches, which have branches. All right, like so. Okay, glycogen, done. Cellulose, not a lot of space. We'll deal with it. Right, remember, its key points, we're not going over every single thing, but cellulose is a beta-glucose polysaccharide. But the key thing here is that cellulose, structurally, the beta-glucose alternates. Every other molecule, no, every other monomer every other monomer rotated 180 degrees. Okay, so every other monomer rotated 180 degrees or flipped over, however you want to call it. But what this causes is straight molecules which are unbranched. Straight molecules which are unbranched, and because of that they can form bundles. Right? They can come close enough to form bundles of molecules which are kind of strengthened by crosslinks, chemical crosslinks, and hydrogen bonds. Okay, so we've gone from structure, and because of that, it gives it a high tensile strength. I'll just put down here strong, but yeah, high tensile strength, because you've got these bundles of molecules form like a rope-like structure, and because of that, they give a lot of strength, and therefore are ideal for supporting the plant cell. Providing the turgor pressure, or allowing turgor pressure in the plant cells, which gives the plant support, prevents plant cells from bursting. Okay, but also, because it's primarily made up of a polar molecule, cellulose is also permeable to water, so it allows water to go in and out of plant cells. Eesh. Right, that's a bit yucky, but it is there. Okay, in some form or other. We did forget disaccharides. Okay, so we've got monomers, but monomers can come together. They can form the polysaccharides that we've got here, but they can also form disaccharides. Okay, so in terms of disacs, right, specifically on the specification are sucrose, fructose, and maltose. Okay, there is something else. We've looked at an example of hexose sugars. Hexose meaning they've got 6 carbons in the ring, but there are also pentoses, which we will encounter a bit more when we do nucleic acids. Okay, so there are this other thing. So we got hexose. So if I just make an arrow like this and call this hexose, we do also have pentoses. Right, so hexoses being 6 carbons, pentoses being 5 carbons, and we will meet them again anyway, but I'll just point them out here. We need to be aware that examples of these that we should know about are ribose and deoxyribose. All right, so that's carbohydrates. We're going to revisit them a little bit when we look at the practical bit, but now we'll move on to lipids. Apologies, I know students hate this, but I have just done a bit of reorganizing of the information. I thought if it's so disorganized, it kind of defeats the purpose of kind of doing this process. So I've organized the information. I'll just quickly run through it again. So we start off with the elements. We put the elements together in monomers. Those monomers can be pentose, i.e. made of 5 carbons. So we've got ribose and deoxyribose. Those monomers can be made of 6 carbons also. Now those monomers include alpha glucose, beta glucose, which we go a lot in depth with, but also include fructose and galactose. Maybe just have a quick look at their structure, okay? But the thing I will definitely encourage you to know is that fructose plus glucose, so fructose plus glucose gives us the disaccharide. Is it fructose? Fructose plus glucose gives us sucrose. Galactose plus glucose gives us lactose, okay? And glucose plus glucose gives us maltose. So alpha glucose plus alpha glucose gives us maltose. So we just need to be aware of the disaccharides as well. It does come up kind of more often than I would expect. But then, yeah, the major part of the discussion is going to be the polysaccharides, which with the alpha glucose polysaccharides I've divided into plant and animals, starch and glycogen, starch comprising amylose and amylopectin, structure, properties, and ultimately function. And again with the glycogen, very similar to amylopectin, only that because of the animal's higher metabolic demands, glucose needs to be released more quickly, and therefore the glycogen is more highly branched than the amylopectin. And finally cellulose, that is, that fundamental structural difference. Every other glucose molecule in the chain is rotated or flipped by 180 degrees, therefore giving us straight molecules which are unbranched, joined by beta 1 to 4 glycosidic bonds, giving us molecules that can come close to each other, form hydrogen bonds, cross-links, bundles, forming strong high tensile strength structures, which can be used as a structural polysaccharide in cell walls. These other polysaccharides are glucose storage polysaccharides. They're not structural, they don't have a structural function. Okay, right, with carbohydrates done then, we'll go over to lipids. Before I go into lipids, I just want to mention here, it's not very clear here. There's a limitation to what we can do, but I think the thing that comes up quite a lot is this relationship, the interrelationship between structure and function. Okay, so for all the functions that you're aware of for a particular molecule, it helps. It's not just know the structures and know the functions. It's important to understand which structural element or which part of the which property specifically is connected to which particular function, like we did with water up here. Okay, so similarly with the carbohydrates, if we're talking about the what is it that makes cellulose, what features of cellulose make it suitable for its function that it is a structural carbohydrate. So the fact that it's insoluble means that it's good at that job. The fact that it has a high tensile strength makes it good for that job. The fact that it's permeable to water makes it good for its job. So just understand those connections. Let's go over to lipids then. So the first major lipid that we look at is triglyceride. Yeah, and remember that a triglyceride triglyceride is composed of the glycerol combined with 3 times fatty acids, okay, and they undergo condensation reaction, and ultimately they produce a molecule that looks generally like this. Yeah, so you've got glycerol, and we've got fatty acid, right? And the bond that connects the glycerol to the fatty acid is the ester bond. We've got the fatty acids. Now, because they're primarily carbon and hydrogen, or not primarily, but exclusively carbon and hydrogen, they are highly non-polar. And because they are highly non-polar, they are very hydrophobic. Okay, and that's going to have important consequences, especially for phospholipids. Okay, so they are hydrophobic. Yeah, and also remember that these fatty acids can be saturated, or they might be unsaturated, making a bit of a kink in the hydrocarbon chain. And has implications for the properties of the molecules. So the saturated can come closer together, the unsaturated with the carbon double bond, they can't come as close together, they can't pack as closely together, which means they tend to be a bit more fluid. Yeah, so there we have our triglyceride, and the main function of the triglyceride is energy storage, right? Because the fatty acids can be used for respiration. Okay, so the fatty acids can be used for respiration. So, well, we can say respiration, and remember that they are more energy dense. They are more energy dense than carbohydrates in that sense, right? So respiration, but also insulation, physical insulation, yeah, as well as thermal insulation, right? So yeah. Okay, yeah. Now, the other kind of lipid is phospholipids. So phospholipids, remember, they are not just completely hydrophobic like a triglyceride. They are they have a hydrophilic head, and a hydrophobic tail, right? Right, so the head is hydrophilic and the tail is hydrophobic, and because of that they can form membranes, phospholipid bilayers, but we discussed that in membranes, so we won't go too much into it right now. But you see how the structure affects the properties, and the properties affect ultimately what that molecule is useful for. Okay, and we also have cholesterol. Yeah, I don't think I'm missing anything. Now, cholesterol, I don't think you're supposed, or I don't think you have to memorize its structure, but it is kind of like a hydrocarbon ring structure molecule, and I think it's got an OH group somewhere like that. So there's a bit of it that's polar, but mostly it's a hydrocarbon ring, it's non-polar. But as you can see, because it's got this hydrocarbon ring structure, it is a planar molecule, which that's why it can regulate the fluidity of membranes. It can insert, so this part here, the non-polar part, can insert and mix and interact with the fatty acid tails of the phospholipids, and this OH group, this polar part, can stick its head outside above the phospholipid or above the fatty acids and interact with the phosphate heads or interact with the water. Okay, so there we have that. I think we just haven't mentioned the elements, right? So for cholesterol, we're simply going to be talking carbon, hydrogen, and oxygen. For triglycerides, again, we are only going to be talking carbon, hydrogen, and oxygen. Not in any particular ratio, however, in this case, unlike carbohydrates. But with the phospholipids, obviously we're going to involve the phosphorus in there, so we're going to have carbon, hydrogen, oxygen, and phosphorus. Okay, and I think that we will leave it there with lipids. Okay, and now we move on to proteins. I mean, we could mention, I guess, that look, these triglycerides are mainly as a fatty acid storage, because fatty acids can be used in respiration by the formation of acetyl coenzyme A. Phospholipids are all about the phospholipid bilayer and membrane formation. Cholesterol are also involved in membranes, but cholesterols can be modified to also produce hormones. Okay, so I will just put that in there as well. Might not have a lot of relevance now for paper 2, however, it might have more relevance in paper 3. Okay, let's do proteins. All right, so proteins are made of, remember, the basic unit, the monomer for proteins is amino acids, and that's where we will begin. But the elements that they are made of are carbon, hydrogen, oxygen, nitrogen, and sulfur. Okay, so the nitrogen is primarily in the amino group, but sometimes it can be in the R group, and there's some amino acids, a couple I think, that have sulfur in them. So these are our elements. Okay, so there are our amino acids. Now, the amino acids, remember, and I won't spend too much time with it, but the amino acids undergo the condensation reaction as well. So know how that works. Just very quickly, just sketch it out here. We have COOH. So if you've got the carboxylic acid of one amino acid, so R group, the hydrogen, NH2 there, and we would have the NH2 of the next amino acid, the amino group there, RCOOH, right? And what's going to happen is that these atoms will combine to form the water, and the C and N connect via covalent bond, forming the peptide bond. So that CN is the peptide bond. Okay, we have produced water. It's a condensation reaction. Cool. Okay, as a consequence of that, we then get the primary structure, and this might have been a kind of throwaway little bit when you first did proteins, but remember the primary structure, it determines, is the primary structure the definition, which I'm not going to go into, but the primary structure is the sequence of amino acids. Define. Primary structure, the sequence of amino acids in the polypeptide, and there are other factors, but the primary structure determines the final folding, what that final folding is going to look like. And the final folding is the shape, and the shape is related to the function of the protein. So if the sequence of amino acids is is different, or if there's a mistake in there, then that will affect the final shape of the protein, which will affect the function of the protein, so that's essential. Okay, and the primary structure is what is determined by the process of the gene sequence, transcription, and then finally translation that determines the primary structure. So when we look at nucleic acids, that story directly connects into this. So don't have it as two separate things, they are part of the same thing. Okay, so the primary structure is a sequence of amino acids. That then takes us to secondary structure. Secondary structure, and remember the secondary structure is all about the hydrogen bonding, right? Because these peptide groups, so after every amino acid there's this peptide group. Of that peptide group, this oxygen is partially negative, this hydrogen is partially positive, and this nitrogen is also partially negative. So those things combine, and peptide groups at different points along the polypeptide chain can interact with each other via hydrogen bonding and produce alpha helix or beta pleated sheet structures. So like mini little folds within the longer polypeptide chain. That's not the final folding, however. We do then have the most important level of the structure. I mean, I don't know if it's, well, it is the most important, and it is the one that has most connections to different points of the specification, because ultimately protein function comes down to the correct tertiary structure, though it can involve quaternary structure as well. So the tertiary structure, remember, is all to do with these R groups. Remember, every amino acid in the sequence has got an R group, and these R groups have different properties, and these properties allow the chain to kind of fold, and these R groups interact with each other, causing the chain to fold in a particular way, determining the three-dimensional structure of the protein. And remember, depending on the nature of these R groups, those interactions can be 4 different types. Therefore, which R group you have at which position determines which interactions can happen, and that determines the shape of the protein, which determines the function. So wrong sequence, wrong R groups, wrong interactions, wrong shape, different function, wrong function. Okay? So tertiary structure is there, and the tertiary structure is all about the R groups of the polypeptide chain, where the secondary structure was more about the peptide group. The R group has, remember, 4 interactions, 4 types of interactions. Do we have the space? Remember the strongest, the covalent disulfide. We've got ionic, we've got hydrogen, and we've got non-polar interactions. Yeah? And remember, the non-polar interactions are all the hydrophobic R groups kind of pushed into the middle of the core of the protein by the surrounding water or cytoplasm, tissue fluid, whatever it is, right? Whereas the hydrophilic R groups tend to move to the surface of the protein, interacting with the aqueous environment. Okay? So that's tertiary structure, but then beyond a tertiary structure, there is quaternary structure. And quaternary structure is all about different, so this is all one polypeptide, but quaternary structure is all about the interaction of multiple polypeptide chains. How do they interact with each other? Well, it's the same story as the tertiary structure. Okay, so whatever interactions can happen within a polypeptide can also happen across polypeptide, because these R groups can interact they're not kind of too selective about which R groups they will interact with. Okay, so we have multiple polypeptides, and that gives us quaternary structure. All right, I don't have a lot of space here, but I think the next thing we're going to discuss is types. So once we've kind of looked at a protein structure, we are going to look at types of proteins, the globular and the fibrous, with hemoglobin as our example of a globular protein and collagen as our example of a fibrous protein. Yeah, so generally we need to know what structural features they have and what properties they have and what functions ultimately that allows them to perform. Quaternary structure then takes us to types. Not that every single protein has a quaternary structure, but types of protein. And in terms of types of protein, we can have two types. We can have globular, right, and we can have fibrous, right? Now the main thing about the globular proteins is roughly that they have a spherical shape. So you know, I'm not going to go too much into it, right? So know generally why globular proteins are globular proteins and what features they have, right? So structurally speaking, what are they, right? Fibrous proteins, again, structurally speaking, globular proteins are spherical, soluble, right? Roughly spherical. They are soluble, right? And they generally have quite a lot of tertiary folds, whereas fibrous, fibrous are insoluble, so they have the property of being insoluble, which generally means, you know, they might have quite a few hydrophobic R groups on their surface, which means that protein needs to find something else to bind to because it can't interact with the water. And usually these fibrous proteins come together because only they can satisfy their binding requirements. It can't be satisfied by the aqueous environment. Right, so they're insoluble, not spherical, form, you know, they kind of polymerize to form fibers, I guess, right? And they have little or very little tertiary folding, so they generally tend to be quite, well, I don't want to say linear, but you know, they're not very folded in upon themselves. Okay, and there we have that. So yeah, our example is going to be hemoglobin. Now let's just quickly remind ourselves, because this is an important idea. Which fibrous proteins are we aware of, and which globular proteins are we aware of? And we are aware of, I think, more than what we might think. Globular proteins are all the enzymes you have ever heard of. Okay, they will be globular proteins. Globular proteins are transport proteins like hemoglobin. Right? You've heard of plasma proteins. Well, plasma proteins are dissolved in the plasma of the blood, right? Therefore, they must be globular. Plasma proteins, enzymes, antibodies, transport proteins, channel proteins, receptors, lots, okay, right? Fibrous proteins, collagen, elastin, keratin. We don't go into too much depth with these, but understand that they have unique properties. and contributions to different tissues in the body, right? Okay, yeah, I will now move on and we'll quickly look at mineral ions, and then it's the practical element for the topic. Okay, I did forget about something here. Globular proteins can, as part of a quaternary structure, so I mean, I like to think of quaternary structure as anything beyond one polypeptide. So one part of quaternary structure is having multiple polypeptides, fine. Another part of quaternary structure might be the idea of conjugation, the idea of having a prosthetic group, something extra, something non-protein, permanently part of the protein structure that is required for the protein to function. Okay, so we're talking about prosthetic groups, which hemoglobin is a good example of that. So prosthetic group, conjugation as being part of quaternary structure. Yeah, okay. So with that said, let's move on to mineral ions. Now with mineral ions, there's quite a few of them, and the list can be a little bit intimidating, but what I would just kind of comfort you by saying is that these are things that have come up in other parts of the specification. It's simply there for you to just make that mental connection to those. So I'm just going to go through each one and just reminding you about where we encountered them in the specification. It might not be an exhaustive list, and I'm sorry about that. But this is the approach I'd like you to take. So if you can follow up by doing your own research to see if these ions come up anywhere else in the specification, that will be to your benefit. Right, calcium. Calcium 2+. Where did we encounter calcium 2+? Remember, the most obvious example to me is any time we need to release or stimulate exocytosis, right? So generally when we've been looking at synapses, we've got movement of calcium ions, and the calcium ions stimulate the release of the neurotransmitter containing vesicles from the presynaptic neuron. Okay, sodium ions. Well, sodium ions come up in a lot of places, but again, the most obvious place is nerve impulses, and generally wherever we've got kind of co-transport situations going on, trying to get molecules to move from one place to another using kind of sodium ion concentrations. Right, potassium. Potassium has kind of cropped up in a number of places. In plant responses, I know, right? Don't mention plant responses, but in plant responses, the ABA abscisic acid stomatal opening and closure is regulated or controlled by influencing the movement of potassium ions. Yep. So we've got that. But remember, also in nerve impulses, we have potassium ions. So nerve impulse, potassium ions in nerve impulse. We've got potassium ions also in stomata, opening and closing. Okay, what else? We've got hydrogen ions, photosynthesis, respiration. I'll just put energy here. Energy, but we're thinking photosynthesis, respiration, chemiosmosis, building the proton gradient, electron transport, all that stuff. Active loading in the phloem, into the phloem as well. And we've got ammonium ions. NH4 plus ammonium. Well, that was mainly in homeostasis and the liver stuff. So I might just put that. Let's just put that there. Liver deamination, but it also comes up in the nitrogen cycle. okay, which is not on the content list for this year, but stuff may come up, you never know, even though its percentage might be not that much, but it might be there. Where else? So now anion, so all these are positively charged, so nitrate, again nitrate, NO3 minus, right, again nitrates, that's in the nitrogen cycle. Hydrogen carbonate ions, HCO3 minus, that was to do with CO2 transport in the blood, remember? Then we got chloride ions, running out of space here, let's just do this here. Chloride ions, chloride, chloride. Chloride ions as kind of cofactors for amylase, so I'll just put amylase down there, amylase cofactor. Is that the only place we've had chloride ions? Phosphate, right, phosphate, that's a good one. PO43 minus, PO43 minus, phosphate, that's come up a number of times. Remember, phosphate groups, think about ATP, think about ADP, it's that phosphate group adding and removing, but also a lot of proteins are phosphorylated as part of cell signaling processes. So I'll just put cell signaling there, involving phosphate as well. Am I missing anything else? And finally, hydroxide. Don't know if this comes up a lot, does it? I mean, we've got protons, so by extension, we've got to have that. I can't really think of OH, so if you think of anything, that's great. I can't really think of OH minus affecting anything. I mean, it is there, it's just not a main character in any story, is it? I can't remember. I can't remember. Okay, well, let's just put that story down as influencing pH. Okay, so just as you have lots of protons and that reduces pH, you've got lots of OH minus and that will increase the pH. That's the only thing I can think of right now. My sincerest apologies. Okay, but if you're not satisfied with that, do look it up, and you know, if you want to put it in the messages and show me up, that would be great. I'm sure other students will find that useful. Guys, thank you very much. Right, that takes us then to the testing. This is long. So the presence, concentration. So let's begin with what we did first. Let's begin with carbohydrates. So in terms of carbs, we could be testing for starch, and starch would be tested for with the iodine test. Okay? We could be looking at reducing sugar, which is what glucose is, and other monosaccharides may also be reducing sugars. And we test for reducing sugars using the Benedict's. Actually, we should talk about the color change as well. The iodine goes from orange, brown. Actually, do I have time to do this? Orange to blue, black. The Benedict's reagent goes from blue to red, and it goes through all the different kind of green, yellow, orange, red. Okay. There is also like a non-reducing version of the test. So you might start off with something that is a non-reducing sugar, but there's a way to do the Benedict's test to even test whether a sugar that you have is a non-reducing sugar. So please look that up. It's a couple of extra steps. I think it's worth knowing. Do the biological molecules have the practical? Oh, they don't. Okay. All right. Presence concentration, carbohydrates, right. And then we could be testing for proteins. No, lipids we did next, isn't it? So the lipids is the emulsion test. Again, know the steps to do the test and know what the correct wordage, phraseology is to describe a negative test and a positive test. Okay, so emulsion test, if it's present, you should get a kind of cloudy white layer at the top if the lipid is present. And in terms of proteins, we do the biuret test, right, which takes us from blue to lilac or purple. Okay, so those are the tests we can do, and those are kind of absence-presence tests, but they can easily be converted to a quantitative version with the addition of colorimeter. All right, because these color changes we can see by eye, and we can make that kind of subjective judgment on whether something's changed from blue to red or not. Okay, but the extent to which that color change happens, that can be measured with a colorimeter, and then that can give us a more accurate, objective idea of how concentrated that substance is. It's not as simple and straightforward as just sticking in a colorimeter, right? For example, with the reducing sugar, it's the precipitate you shouldn't really be putting that directly into a colorimeter because the precipitate is a solid. So ideally you want to be removing the precipitate and you want to be measuring the absorbance of the remaining blue solution. Okay, the less blue it is, the more reducing sugar there was, because the more reducing sugar there was, the more of the blue copper ions have been reduced to the red version of the copper ions. Okay, and the more copper ions that have been converted into the precipitate or the solid form, the less blue the solution will become. Okay, so if that didn't make sense, it's not really the point of the video, but if that didn't make sense, like do look that up. This is me telling you, signposting you to like look that up if that didn't kind of settle well in your brain. Okay, but know that there's a basic version, the qualitative version, and know that there's a quantitative version, and it's very, very useful to know both of those. Right, similarly with the other ones, you know, you can, I don't know how you do it with the emulsion test, but yeah, you could do it with the proteins as well by using a colorimeter to judge the extent. Now, if you do use a colorimeter, we don't have time or space to discuss it right here, but you would, by itself, the absorbance value doesn't tell you anything unless you have pre-made a calibration curve using known concentrations, right? You use a range of concentrations, perhaps with serial dilutions, and you make a calibration curve, and then when you read your test value, you simply read off the absorbance value of your calibration curve, and that will tell you how concentrated the solution is. Okay, so that's that. I hope I'm not missing anything. But then that takes us to separation, and that is more to do with the proteins and amino acids and perhaps even like chlorophyll molecules. Okay, so the idea of separation is really chromatography. Now this, if it does, I mean, this does tend to come up a lot. So, you know, do understand the principle, do find exam questions to do with chromatography and make sure you're kind of practiced in those. But the idea of chromatography is that you're taking a mixture. So you've got in a beaker, you might have a solvent, right? And in the solvent, you're placing your substrate, could be a piece of paper, could be some kind of membrane, but on that you have dotted mixtures of substances, yeah? And the solvent has a different capacity of, because the mixtures of molecules, each of the molecules has a slightly different property based on slightly different structures, they'll all have a slightly different tendency to get dissolved in the solvent. So as the solvent permeates through the paper, right, as it gets drawn up the paper, it dissolves the molecules within the mixture, and carries them up the paper, and depending on the properties, depending on how well each molecule dissolves in the solvent, they get carried to different extents. Okay, so the mixture can be separated. Now, these different molecules, so this, you know, the molecule A moved up this much, molecule B moved this much, molecule C moved that much. The way we can compare them is by using the RF value. RF stands for retention value, or what is F? Okay, retention factor, whatever, okay? And is the ratio between the distance moved by the solvent, so the solvent front, so the solvent's going to be somewhere up here, okay, the distance moved by the molecule C, for example, divided by the distance moved by the solvent, and each of C, B, and A will have a different RF value as a result of that, because they each moved different distances, and that can be used to kind of identify them, differentiate them, etc. Oh, right, biosensors. Right, so the concentration can also be measured, so you can do it via a colorimeter, but a more direct way and quicker way is biosensor. So it's an electronic device. It converts the presence or absence of these molecules into an electrical signal, which gives us a reading and a value. That's it, I think. Okay, guys, that was long, but it is there. Hopefully, again, look, if I've made mistakes, sincerely apologize, but check it. If you have any doubt over anything that I've said here, please do check. There's a lot of information. I might have made a slip up somewhere, but it's hopefully you get that this is being done with the best of intentions. Right, so don't take my word as gospel. Please do check. If you have any doubt over anything that I've said, please do check it. And where I've encouraged you to kind of go beyond what's here, do that as well. There's only so much you can fit on a board like this. So yeah, I mean, there's a lot of stuff there's quite a bit of stuff that I've only pointed towards, but this is the broad strokes version of what you need to know. Okay, so everything that could be in the topic should be here, if not all the detail of it. Guys, I hope this has been helpful, useful. I will now start working on the next video. Good luck, guys.